Professor Joseph Sung. Many of us that have been involved in gastroenterology will be aware of lots of Joseph's work, starting off with Helicobacter, but moving into literally practice-changing, worldwide practice-changing research on peptic ulcer and how to manage it, and thereon into the management of colorectal cancer, especially around the early detection and screening. Of course, he's also extremely well known for the response to SARS, which was the pandemic which hit, or the epidemic that hit Hong Kong back in the early 2000s, and for that he's been recognised with numerous awards. Joseph is a great friend of the University of Oxford. He's been here before, I remember very recently, and is a huge friend of the Centre for Personalised Medicine. So it's with an enormous pleasure and a great gratitude to him to welcome him to Oxford again to speak on gut microbiota in GI cancers. Joseph, thank you very much.
Thank you very much, Simon, and everyone who comes here. It's a sunny, beautiful day outside, so you should be in the sun instead of coming here. But I'd like to thank Simon for his invitation, and it's really my great surprise to see Professor Derek Joel and Professor Richard Hunt here. We know each other for only a few years, but since I was a teenager, and in fact, Richard, you probably don't know this. 35 years ago, when I was doing my PhD, you were invited to be the examiner for my thesis, but you didn't come because of something else, and so I'm going to present to you my PhD thesis right now. And that is really on the bacterial biofilm, but I twist the topic a little bit because there is a lot of development in our understanding of gut microbiota and GI cancers. In fact, if you attend the DDW this year, and I presume the same for British Gastro Society's meeting, about 1/4 of the paper is really on the microbiome. So it really is a paradigm shift and revolutionized a lot of our understanding of these diseases. We all know that we are all surrounded by microbes of any kinds, including bacteria, virus, and fungus. But in fact, the GI tract is the biggest residence for many of these microbes. And it has been estimated that there are more microbial cells in or on our body than our own human cells. So when we say we actually talk about our human cell as well as many microorganisms surrounding us. And it's been estimated that they weigh about 1.2 to 2 kilograms, and the metabolic output equates to what the liver is doing in our body. So these are our allies in many ways. We are living in a symbiotic conditions. They benefit us and we benefit them. But the bacteria is doing really a lot of things for our human body. It helps us to digest the food, including carbohydrates and also a lot of fibers, and production of short-chain fatty acids, which is extremely important for our health. And also, very importantly, now we start to realize that the metabolism of bile acid by microbial organisms in our body is holding a very crucial role in the cell proliferation of our GI tract. Urease activities to balance the nitrogen content of our body, synthesis of vitamins, and also metabolizing the drugs, various kinds of drugs that we are taking every day. But here, I'm here in the last 2 days to learn a lot about GI immunology, and I think the microbiome in our body has a lot to do with our host's innate as well as acquired immune system, and that translates into a lot of conditions that we are interested in. But in the past 2 days, when I talked to colleagues in Oxford, I've talked to actually students from Russia, I talked to postdocs from Italy, people from Germany on the faculty, of course a lot of Brits here, Indian faculty members. So this is really a very diversified community, and diversity is good because every nations, every kind of bacteria, I'm not saying that you are bacteria, but diversity is actually good since if we can coordinate well and each contribute their strength, it will give health to us and resilience in our body. And that is the same for our microbial communities in the body. One of the examples that we can see is that when we eat our diet, our bacteria actually helps to digest the fiber, and that keeps our intestinal tract healthy because it will maintain the mucus layer, and that will try to avoid pathogens in direct contact with our epithelial cells. This is really my research thesis 35 years ago on bacterial biofilms, and we understand that the passage bacteria on the surface don't just pass by, some of them will stay, and they're well protected in the mucus layer in a symbiotic relationship with us. And if this ecology is being disturbed when the mucus layer is gone for any reason, we will be under direct exposure to pathogens, and that upsets the homeostasis of our body. So in Hong Kong, our group has been interested in studying colorectal cancer from pathogenesis to finding genomic markers and epigenomic markers for the diagnosis in the past 10 years or so. But in the last few years, we have really shifted our interest to study metagenomics, including gut microdysbiosis, because we start to believe that this is playing a crucial role in the diagnosis as well as treatment for colorectal cancer. So these are some of the publications that we have to put out. In other words, we shift our attention from genomics to microbiomics. And Simon talks about personalized medicine, and we have a lot of focus on the genomics and epigenetics of the host. But I believe that when we talk about personalized medicine, we don't just look at the host genomics. We also have to look at what are we harboring in our body, the trillions of microbes that is part of us, and that is the microbiomics that can produce health or upset our health if it is not in a good condition. And there are many mechanisms. We still only understand a very, very small part of it. From the dysbiosis, we know that it will stimulate our immune system, triggers inflammation in the body, and it causes perturb the metabolism of our body. And one of the examples is on bile acid metabolism, which in the end can cause damage to the gastric epithelium, and also triggers the direct toxicity to epithelial cells. So what I'm going to try to discuss with you is on these four areas. First of all, our observations of dysbiosis in colorectal cancer. And are these changes really the cause or the consequence of the malignant process? How can we utilize this understanding in making early diagnosis, and how can we perhaps use this as a preventive measures? There should be also a lot more data on how that could affect our treatment of cancers, but I'm no expert in this area, and we have known only very little about it. First of all, on the microbiomes, we believe that there is altered gut microbiota composition in colorectal cancer, and I'm going to show you some of our observational studies. And that is not just on cancerous patients, but also looking at in the process of cancer development at different stages from pre-malignant to malignant lesions, how the microbiome is being affected, and also what role they play in the functionality of this microbiota. So we start with looking at a group of patients with colorectal cancer and finding a group of controlled patients. And the patients, we use the stool to study our microbiomes. I apologize for this. We look like a chocolate ice cream, but this is the best picture I can find on Google to represent the stool. And we know that this is not the best specimens to understand the microbiome because they are, in fact, the transit bacteria and virus. we should also look at the sessile population, which stays on the surface of the epithelium. And we used the shotgunned metagenomic approach, and we found that comparing to normal individuals, there are a number of phylums which are particularly enhanced or enriched in patients with colorectal cancer. And these are a few of them, and you are all familiar with Fusobacterium, Nucleatum, and Parvimonas, Peptostreptococcus, as well as a bacteria which you may not be so familiar with is Solobacterium. They're all found enriched in colorectal cancer patients. But all in all, in fact, the diversity of the microbiota in the gut is shrunken in cases of colorectal cancer patients. We also found that among the 5 enhanced bacterial species, 3 of them are actually oral pathogens. And in fact, 2 of these, including Parvimonas and Fusobacterium, they're actually found in the infected root canals of our teeth. And therefore, oral commensals or oral pathogens has a lot to do with what we may be suffering from if this bacteria goes into our gut when the normal ecology is being disturbed. But we're still not sure what is the role played by such organism. And these organisms are not just coming in and going out on their own. They seem to have a network. They seem to come and go together. So we observed that there is a co-occurrence of 21 species in colorectal cancer patients compared to the control. And they are listed here. And among them, again, oral pathogens play a very important role. And there is also positive as well as negative correlation. That means if one bacteria disappears, the other one may also disappear at the same time. So they go hand in hand in both directions. And we try to use this as a marker to identify who are the patients that might actually be suffering from colorectal cancer. And 20 gene markers have been identified using the minimum redundancy and maximum relevance technique. We identified 20 gene markers that separate, and you can see here that they are much more common in colorectal cancer patients than control. And we also use other diseases such as type 2 diabetes and IBD patients as a control, and they seem to be quite specific found in colorectal cancer. And this study used 51 cases compared to controls. We found that at every stages of colorectal cancer, from stage 1 to stage 4 disease, these gene markers can be found. And if we put a ROC curve, they seem to be fairly useful in identifying the patients who have colorectal cancer. We know that FIT test is the best stool test so far and widely used for detection of colorectal cancer. But this is only when the tumor is bleeding and hemoglobin is being picked up. So if there is other parameters in the stool that we can use to detect colorectal cancer, maybe we can add to the FIT to improve its sensitivity and specificity. So this is validation of our markers using a group of patients from Denmark. Although the numbers are small, but the ROC curve still seems to hold up for the diagnosis of colorectal cancer. And we subsequently have tested it in Austria and France as a diagnostic test. Next question we ask is that, yes, we found this bacteria. Are they the chicken or the eggs? Is it that the story starts from dysbiosis and then change in metabolism, development of adenoma, eventually colorectal cancer? Or in fact, it is the cancer environment which facilitate or enhance the proliferation of this bacteria. So which is the cause, which is the horse, and which is the cart? And that is the question we want to ask. In order to answer this question, we have done another study looking at different stages of colorectal cancer development. And there are patients who have cancers, patients who have adenomas and control, and we do the pyrosequencing of these patients. And the different colors here represents different stages of the disease, and this is representing cancer. and there are cancer-adjacent tissues over here, and there is a normal control. And this big one is the adenoma population, and this is adjacent tissue to the adenoma. You can see that they seem to represent quite distinctly different meta-communities. And you can see we can group them together as 5 different groups or meta-communities representing different groups of bacteria. And they are found in quite distinctly in different pathology or the adjacent normal tissue next to the pathology. And this, we believe that may be useful as a mucosal phenotype prediction. And in fact, when we plot them separately into 5 different organisms, and you can see from normal to adenoma to cancer development, the concentration or the population of these 5 different kinds of bacteria goes up. And it seems that they are not only found in advanced cancer, but they can also be found in pre-malignant condition, as well as to a certain extent in the adjacent tissue next to the cancer. So it seems to be an early process, an early colonization instead of colonization after the malignancy is settled in. And once again, we found that there is some relationship between the microbiomes from the normal tissue to become adenoma to become cancer. And this complicated curve shows you that there is, in fact, a discordance of microbiomes from cancer to adenomas. In fact, when the patient developed cancer, the discordance between the lesion and the lesion adjacent to the tissue is greater. The thickness of this arrow represents how many % of discordance between the microbiomes in the lesion and adjacent to the lesion. And it is more pronounced in advanced cancer than in polyps with low-grade or high-grade adenoma. So that tells us more and more about the interactions. You see, in a normal tissue or the normal person, this is the kind of positive correlation between the organisms. And then when it goes to the adenoma and carcinoma stage, we see this co-occurrence seems to drop, but there is a lot of co-exclusion of bacteria or microbiomes that we can identify using pyrosequencing. And we believe that this may implicate a change in the host environment that facilitates certain type of bacteria to coexist or they disappear together. And this response may also shed some light on the change in mucosal microenvironment that could explain the development of cancer. And this environment could really be inflammation induced by the bacteria and enhances several types of bacteria. We know that one of them, actually Bacteroides fragilis, is a inducer of signal transduction of Th17 dependent pathway, and then the other has already been identified as one of the organisms associated with pancreatic cancer. But this is still not fully explaining whether this could just be a coexistence or whether this is the consequence of inflammation and cancer development. So in order to answer that question, we did an animal study by feeding the stool of cancer patients versus control patients to the animal, and we add a carcinogen to facilitate development of adenoma. So this is a animal study that we treat the mice and remove the microbiome antibiotics, adding AOM to it, and then start feeding them with the stool. And you can see that with time, the mucosa developed from normal to low-grade to high-grade dysplasia, much more so when the animal were fed with stool from colorectal cancer patients than stool from a normal individuals. And the time shows that the proliferation of cells indicated by Ki67 increases, and you can see the crypts start to convolute and become more and more developing into dysplasia and cancer. Now, why would that happen? When we look at a a cascade of inflammatory cytokines, we found that these colorectal cancers do upregulated expression of at least 33 transcripts of inflammatory pathway, including Th17, IL-23, and also attracts monocytes, T lymphocytes, and other inflammatory cells. So it does relate to the inflammation of the gut. And this is just to put it in another way. You can see these cytokines are being upregulated when the animal were fed with colorectal cancer patient's stool. On the other hand, it also upregulated 37 transcripts of cancer-related genes. Some are related to angiogenesis, some correlate to invasion and metastasis, cell proliferation, stemness, apoptosis. And here are the 37 genes that are upregulated as an oncogenic cofactor. And we believe that the stool has actually directly or indirectly work on this gene, leading to increased carcinogenesis, angiogenesis, and metastasis of the tumor.
Is this the small intestine of the mice or the lower?
These are the large intestine. Yeah. So they develop more polyps, and the tissue shows more inflammation and more oncogenes being turned up. And one of the bacteria that we have go into more detail is the Streptococcus anaerobius, and it's found that on top of inflammation and oncogenes, it also turn on the, it causes high-grade dysplasia in the intestine. As you can see, this is the large intestine, and with the carcinogen, E. coli doesn't have that much of effect, but when you put this Streptococcus, you see a lot more polyps develop and with dysplasia. And that, I won't have the time to go into the details, but it actually goes through a toll-like receptor pathway and causes increase in intracellular cholesterol biosynthesis, and which may affect cell proliferation, and hence maybe at least part of the reason why it can cause development of colorectal cancer. And if we again use this kind of organisms as a biomarker for the diagnosis of colorectal cancer, we found that each one of them, particularly the Fusobacterium, are actually quite distinct in cancer patients compared to control patients. And this is the ROC curve, and you can see that compared to control, these are fairly remarkable in terms of its sensitivity and specificity in detecting cancer. However, it is not perfect because the sensitivity rate is only about 80-something%. So we confirm this finding using two different cohorts. Some are better than the others. But what we have also tried is to add FIT test, the FIT test, on top of the biomarkers using this microbiome as a marker. And you can see that without any microbiome marker, FIT can only reach the sensitivity of 40% in a stage 1 disease, but it goes up to over 70% when the 4 markers are added on. So it seems that we may be able to add something to the FIT test to enhance its sensitivity and specificity for the diagnosis of colorectal cancer. So up to this stage, we believe that these changes, this dysbiosis is genuine, and it's found consistently in colorectal cancer. And the progression also go hand in hand with the stepwise progression of carcinogenesis. We believe that the stool, something in the stool, the driver bacteria, actually promotes colonic tumorigenesis, and maybe we can use this marker in the future diagnosis. However, what can we do to prevent this from happening? Well, we know that what we eat is actually affecting a lot on our gut microbiome. So if you go to Mars and still buy McDonald's, you will get the McDonald's microbiomes very soon. So a lot of studies on the diet and the gut microbiome has been published, and this is one of the earliest studies which used two different types, very different types of diet, the plant-based and the animal-based diet, and see how it would affect the microbiome. Of course, when you have plant only, you have a lot of fibers, less fats and less protein compared to the animal-based diet. But more importantly, when people are fed on plant diet, there will be a short-term consumption of this diet actually alter the microbial community structure. In particular, when a lot of animal fat and proteins are being ingested, it will increase the abundance of biotolerant microorganisms. And these organisms seems to have something to do with the inflammation of our gut. On the other hand, the decline of other organisms, such as Firmicutes, may actually be detrimental for our gut. And this is a further study on the diet and fat induced taurocholic acid, one of the biosalts, and found that it can actually promote the colitis in a certain certain mice which are prone to have inflammation. So this diet high in saturated fat, but not so high in polyunsaturated fat, is no good for our gut because it is actually pro-inflammatory, particularly in susceptible individuals. And not just about the nutrient in the diet, but also the additives in the diet may have a lot to do with our gut microbiomes. So these include emulsifier agents and thickeners, which contains mostly carboxymethyl cellulose and a lot of other food additives such as sweeteners and so on. But what is emulsifier doing in our food? I can tell you all the pretty stuff and all the tasty food actually contained emulsifier. Ice cream, cakes, yogurts, French fries, and so on. So try to remember this tonight when you have your dessert. Because the emulsifier, which contains these substances, actually disrupt the microbiota and induce low-grade inflammation. And of course, they have other metabolic effects. And in patients with IBD, this has been suspected to be one of the reasons for recent increase in IBDs around the world because of the use of these substances in our food. So dietary emulsifier was found to have direct effect on human microbiota on the gene expression potentiating intestinal metaplasia, intestinal inflammation. Diet is one thing, exercise is the other. So this study from England, actually it's an international study, shows that people who have exercised a lot and on extreme diet, they have tremendous effect on the gut microbiomes. And I know that the English team has won one of the games in the World Cup, so I've deliberately put up this, this picture here to cheer you up. And it would be nice to have the stool collection before the game and after the game and see how much changes has been produced in the gut. Because exercise alone seems to be able to increase Akkermansia, and Akkermansia is known to be a mucinophilic bacteria that reside in the mucus layer, and therefore they have protective effect. And the reduction of Akkermansia in people who doesn't do any exercise may have a changed microbiome. Finally, since Richard is here, I have to say something about Helicobacter pylori. And this is our old friend, not so old though, Barry Marshall, who was the first one who rediscovered Helicobacter pylori in the stomach. He won a Nobel Prize for this discovery, and he even braved himself to swallow the organism into his own stomach and perform endoscopy to demonstrate that it causes gastritis, try to fulfill the Koch's hypothesis. But then a lot of changes have happened because we have been working very hard to try to eradicate H. pylori, and therefore we can be able to stop progression of atrophic gastritis. Peptic ulcer disease has been almost vanished, and that's why gastroenterologists like myself have to do more boring jobs like a vice chancellor. And but we still have gastric cancer that is still remained a major health problem worldwide. And this Correa's model of gastric carcinogenesis is familiar to all of you, from chronic gastritis to develop into gastric atrophy, intestinal metaplasia. We know that this process takes a long time from normal to dysplasia and cancer. And if we remove this bacteria early enough, perhaps at the stage of chronic gastritis, we may be able to stop the process of gastric carcinogenesis. However, is that by itself sufficient to stop the process of gastric carcinogenesis? Is there a role of dysbiosis in the development of gastric cancer? So we try to look at this by studying a group of patients who has gastric cancer and compared to normal people. We take a biopsy of the stomach and look at the mucosal microbiome. And we compare patients who have superficial gastritis, atrophic gastritis, intestinal metaplasia. That means every steps in the process of gastric carcinogenesis and gastric cancer. And we compare different sites of the stomach for the microbiome pattern using pyrosequencing. And when we look at different sites for the same kind of lesion, that is superficial gastritis, atrophic gastritis, intestinal metaplasia, or gastric cancer, when we biopsy the antrum, the body, and the fundus, and look at the microbiome pattern, we did not see a significant difference wherever you take the biopsy. So there was no topographic huge differences there. But when you compare different pathology from superficial gastritis to gastric cancer, you see a significant difference in the microbiomes. When you compare atrophic gastritis to gastric cancer, you see a significant difference. And so for the case of intestinal metaplasia. So it depends on which stage that the stomach has gone into and not which location that you biopsy that you will detect a different pattern of microbes. And this is a picture to show you comparing gastric cancer with superficial gastritis. And then this is the spectrum of microbes that we've studied. And you can see from the color here, they are very different. In particular, I would like to point out again that among the 21 enriched microbiome in gastric cancer patients and 10 which were depleted, actually there were quite a few of them, again, coming from the oral commensals. So the oral organisms somehow play a role in the gastric, in the process of gastric carcinogenesis. Once again, the correlation of bacterium is found in patients with superficial or atrophic gastritis. The green lines represents co-occurrence, and the red lines represents co-exclusion. So these are the 5 most common organisms we found in stomach of the gastric cancer patient, and there are strong correlations found in patients with atrophic gastritis and gastric cancer compared to superficial gastritis. And if you use this as a marker, the gastric cancer enriched organism as a marker, you may also be able to predict which patients may develop, may have gastric cancer developing. We are still in the process of studying what Helicobacter is doing in the interaction of gastric microbiomes. And in this study, we found that HP negative subjects have less interference on the microbiome interaction compared to the HP positive subjects. So what does it mean? We are still in the process of studying this. But what we know is that the stomach is also affected by a lot of other external factors, including many drugs that people are taking every day. And that includes not just antibiotic, but a lot of non-antibiotic drugs that have extensive impact. In particular, all the PPIs that we prescribe to our patients with peptic ulcer disease, with dyspepsia, with reflux disease, and they are shown to have a broad anti-commensal activities directly influencing the gut microbiome composition. And that includes every kinds of proton pump inhibitors. So what does it do with changing the microbial environment of our stomach as well as the intestine? Will they enhance the risk of cancer development? Will be a very important subject, and it has already been published by several groups of people that PPI altered the composition of gut microbiome in both HP positive and HP negative subjects. And this is a twin study from Jackson and colleagues finding that if you have been giving patients, twins, healthy twins, PPI with one and no PPI with the other, it will significantly reduce the abundancy of microbial diversity among those who received PPI. And there's abundancy of oral and upper GI tract commensals found in these subjects as well. So I think, and there are also studies which shows that metformin, aspirin, and a lot of other drugs actually can alter our gut microbiome. So that would be a very interesting topic. I would perhaps stop here and finish by acknowledging my colleagues. And these are the team of our students and postdoc fellows. And this is our colleagues in Chongqing, in the 3rd Military Medical University in mainland China, who do the germ-free animals for us. And we are partners in this state key lab of digestive disease. And my purpose in here is really to try to explore. My purpose here is to learn from you guys about inflammation and the gut, and I try to establish some collaborations with you in the future. And I hope that our understanding of microbiomes and your understanding in genomics, in immunology of the gut, can enhance each other for the future studies. Thank you very much.